looking at the GFR and mechanisms of regulating GFR. And we talked about 3 intrinsic mechanisms that kept GFR at 125 as long as the mean arterial pressure was anywhere between 80 and 180 millimeters of mercury. Okay, oops. And so let's go over what a response to that prompt would look like. Okay, before I kind of generate a response here, does anyone want to share their response? Does anyone want to share their answer that they gave for this question? And I'll type out the question here in the meantime. So this was our prompt question. Okay, and I don't see anyone sharing. So I will post a response that I generated, and we'll kind of go through this response and talk about what's important, okay? Okay, so we talked about 3 mechanisms of regulating GFR. And I'll probably post this as a screenshot in today's recordings as well, so you can have access to this. So we know that GFR is controlled by 3 mechanisms that are intrinsic to the kidneys, meaning that there are no extrinsic, no hormones, no neurons that are involved. And of those 3 mechanisms, 2 of them alter the afferent arterial resistance. And when we say afferent arterial resistance, we mean the diameter or the size of the afferent arteriole by playing around with the state of the smooth muscle that lines the arteriole. And when the afferent arteriole constricts, we know that the pressure inside the glomerular capillaries decreases, and that's how we can affect a change in GFR. So we want to keep GFR constant in the face of changing our mean arterial pressure or changing the pressure that's coming in. So the first of those mechanisms that involves arterial resistance, right? The first of the arterial-related intrinsic mechanisms is called myogenic regulation. And this is basically an increase in stretch that is seen when the pressure increases that results in afferent arterial constriction. So whenever there is increased pressure coming into the arteriole, it's going to stretch, very similar to that baroreceptor reflex we talked about. And that is going to result in a response of constricting of the smooth muscle in that arteriole. That constriction tends to limit the change in pressure within the glomerular capillaries, and that is what keeps GFR constant. So that's the first mechanism. Stretching of the afferent arteriole results in constriction of the afferent arteriole. The second arteriole-related mechanism is called tubuloglomerular feedback. And this is where the afferent and efferent arteriole, so both the vessel that is bringing blood to the glomerulus and the blood that's taking blood away, come in contact to a distal part of the tubule, hence the term tubuloglomerular, right? These 3 structures are coming together. And remember we said that this had some controversy about whether it was the ascending limb, the descending limb, or the distal convoluted tubule. So for our purposes, we're just gonna say the distal tubule, okay? And any increase in GFR results in increase in flow of filtrate through the distal tubule, and that makes sense. If we're filtering more blood through the glomerulus, we're gonna have more filtrate running through the tubular structures. So the macula densa, which is a sensory group of cells in that distal part of the tubule, they're going to be sensing any changes in the flow of the filtrate coming through. Okay, and their response to increased flow is to release paracrine factors that cause the afferent arteriole to constrict. This would again decrease the pressure of the glomerular capillaries, hence decrease GFR. And in this instance, the flow of the fluid, which is also a product of GFR, I keep saying GFR, through the distal tubule would remain constant. So just to kind of summarize those first two mechanisms, they're kind of doing the same thing. They're causing the afferent arteriole to constrict, but the stimuli are different. For the myogenic response, for myogenic regulation, the stimulus is the stretch of the afferent arteriole that causes the constriction. And for the tubular-glomerular feedback, the stimulus is an increase in the flow of filtrate that's moving through the tubular structures. And then that is relayed via a paracrine or chemical signal to cause constriction of the afferent arteriole. The third mechanism we mentioned is not involving the afferent arteriole at all. In fact, it involves a group of cells called mesangial cells that are surrounding the glomerular capillaries. So these are inside the glomerulus itself. And so an increase in pressure of the glomerular capillaries causes these capillaries to stretch. And then the mesangial cells that are located around the glomerular capillaries are then going to constrict in order to decrease that flow. So the constriction makes the radius of the glomerular capillaries smaller, and this decreases the GFR. So that third mechanism, to kind of summarize that, stretching of those mesangials, stretching of those glomerular capillary cells causes the mesangial cells that are around them to constrict, and then that brings in the radius and decreases the GFR. Okay, so those three mechanisms help to keep our GFR constant in the face of changing mean arterial pressure, as long as it's within that window of 80 to 180. Okay, this is what a essay type response to this question would look like. Okay, just kind of describing each of those mechanisms and talking about how they relate to GFR. All right, any questions on that? Any questions here? Okay, let me clear this. Okay, so this image is depicting the juxtaglomerular complex. Now, I'm not going to ask you guys to know too much about this. This is quite detailed, but I'll talk a little bit about it as it applies to the tubuloglomerular feedback. So that second mechanism of intrinsic control. So these are the structures that are involved in that paracrine signal. So this is that distal part of the tubule that's coming in contact to the efferent and afferent arteriole. And this is how that communication is transmitted. So the cells here, which come together to form this complex, the macular densa cells and these other two types of cells, which you shouldn't really be worried about, basically pick up increase in the flow in the tubule. So increase in the filtrate here and send signals to the afferent arteriole to constrict. And so that decreases how much blood is coming in, how much blood is being filtered, and essentially how much filtrate ends up in the tubular structures, okay? So this is just kind of the complex that's responsible for the tubuloglomerular feedback. Again, I won't ask you to know too much about the details here, just basically the overall response that's brought about to lower GFR. All right. And we went through this flow chart, I think last time we met. This goes through the myogenic regulation and then the tubuloglomerular feedback. And here we're going to pick up by looking at the extrinsic mechanisms of control. Now, we go back to this initial graph that we started at, and we talk about where the extrinsic control of GFR comes into play. So the intrinsic mechanisms are really in this zone, right? So 80 to 180, and that keeps the GFR at 125. But if we drop our mean arterial pressure below 80, or if we increase our mean arterial pressure above 180, our extrinsic mechanisms come into play. Okay, I'll say that again. If our mean arterial pressure goes below 80 millimeters of mercury or above 180, our extrinsic mechanisms come into play in order to control or regulate GFR. So this would happen in the instance of decrease in blood pressure. So that's going to be the more detrimental or more important response. If we decrease our mean arterial pressure, that can be brought about by things like shock, hemorrhaging, dehydration, or excess sweating. And if we drop our mean arterial pressure below 80 millimeters of mercury, there are two ways that we can influence GFR. So there's going to be a direct way, which is if we decrease mean arterial pressure, we decrease blood coming to the kidneys, we decrease our GFR just directly. And that makes sense. Less blood coming through the body, less pressure, less blood coming into the kidneys, and less GFR. So that's a direct correlation between decreased mean arterial pressure and decrease in our filtration pressure, or GFR. But there's also an indirect mechanism that involves the autonomic nervous system. So the indirect mechanism we'll talk about on the next slide, but that is also going to influence decreasing GFR as a result of decrease in mean arterial pressure. And as you can see at this proximal part of the curve, that we no longer keep GFR stable at 125 if our mean arterial pressure drops below 80. In fact, it's going to be imperative for GFR to drop if our mean arterial pressure drops below 80 as a protective mechanism of our blood volume, of our blood pressure, and just a protective mechanism of life, okay? We're going to have to drop GFR if mean arterial pressure gets anything below 80. Okay, now I'll explain how that happens here. So this is another really nice flow chart. This is talking about an instance of hemorrhage or excess sweating. And what happens here is that we lose blood volume. We lose blood volume, and that drop in blood volume is going to drop venous return. Now, immediately we should be making a connection here about our baroreceptors. That is going to help with this response. And so drop in venous pressure causes a decrease in venous return to the heart and a decrease in cardiac output, okay? We should be thinking about the cardiovascular system here as we make this connection. If I've got a lot of blood loss, I'm losing blood, or I'm sweating profusely, my venous pressure drops, less blood comes back to my heart, and therefore less blood is going to leave the heart. So stroke volume decreases, heart rate decreases, therefore mean arterial pressure decreases. Now, this is going to bring about a direct drop in GFR. If my mean arterial pressure drops, there's less blood moving through my system, there's less pressure moving through my vasculature system, and so that would directly decrease my GFR. But there's also an indirect relationship here. And so you can sort of make a note in your slides that this would be the indirect response that's brought about using the autonomic nervous system. So our baroreceptors are going to detect that drop in mean arterial pressure. Remember, baroreceptors that are in various parts of our vasculature, specifically our venous system. We've got baroreceptors in the carotids, and they're going to detect a drop in mean arterial pressure, and they are going to elicit the sympathetic nervous system, which will send those sympathetic signals to the kidneys. And the kidneys, under sympathetic innervation, are going to vasoconstrict both the afferent and the efferent arterioles. So I'll say that again. So if we drop our mean arterial pressure, our baroreceptors pick up that change, right? Remember, these are stretch receptors. They're going to send signals to the brain, right? Our integrating centers, which will send out sympathetic influence or sympathetic innervation down to the kidneys. And under the influence of sympathetics, the kidneys are going to vasoconstrict both the afferent and the efferent arterioles. And we want to distinguish this from the intrinsic mechanisms. Remember, the intrinsic mechanisms, two of them only constricted the afferent arteriole. Here, with the extrinsic mechanism, we're constricting both the efferent and the afferent arterioles. And this constriction is actually going to decrease how much blood is flowing into the kidneys. So we increase our vascular resistance of the kidneys. That will drop GFR. Remember, if we're talking about resistance here, we're talking about the diameter of these vessels. And so if we increase resistance, we're essentially decreasing the radius. So less blood is flowing through the afferent and the efferent arterioles, And so that would mean less blood is being filtered, and that would mean GFR is decreasing. And when GFR decreases, our urine flow decreases, and so does our fluid loss. So we're essentially going to lose less fluid as a protective mechanism of excess sweating and excess hemorrhaging. And this is what I mean by saying that if mean arterial pressure goes below 80, we must drop GFR really in order to stay alive, okay? And if your mean arterial pressure is below 80, that's really a dangerous zone. And so losing excess fluid via urine is not an efficient mechanism. You need to drop GFR to keep urine output low in order to maintain your blood volume and to maintain your blood pressure. So that's a protective mechanism.